Welcome to Journal Pub. We're still not in an actual pub. We still haven't figured this out. We've got wine today.
Thank you to Megan and Zach for always hosting.
I'm pretty nervous about this one because the previous two that we've done, I've known something about, and I at least printed out. I didn't print this out, Megan printed this out, so this is the first time that I'm seeing this. No idea what's about to happen. And the only thing I know about this paper is that I saw a Reddit post about the cover figure.
Okay.
Because people thought this was a pretty funny cover figure.
No, that's why I picked it solely based on the graphical abstract, and then I sent it to Bez and I was like, we should do this for Journal Pub. And then he was like, this paper is not very friendly to my which I felt bad about. I did give him the opportunity to veto and pick a new paper. So I did feel bad about that, but I did actually read this paper because I was like, okay, I feel like maybe it would be good for at least one person to have a little bit more of an idea of like the flow of things leading up to it. We'll see how it goes. If we want to go back to just like blindly like delving into figures, I think that's fine too.
We got some shit for the Planaria one that people are like, you guys don't know anything. So we just want to, disclaimer. Does anyone think that they have a good disclaimer that they can say? Otherwise I'm just going to rant for like 60 seconds.
Go for it.
Yeah.
Okay, this is kind of like a vignette of what it looks like when scientists are encountering material for the first time. We're trying to make it not super slow, because like what we would normally do if we ran into a word that we don't understand is we'd go research that word. That's incredibly boring to watch, so we just kind of power through, which you sometimes do when you're looking at something for the first time. We're trying to make this lighthearted. We've picked, if you happen to be on the paper, we've picked your paper because we're interested in your work and we're dedicating a little bit of our lives to something that you dedicate a lot of your life to. We know the struggle. We're sorry when we get things wrong or when we're critical, but also like this is what it means to engage with your work, right? So to some extent, I hope that people are happy about this kind of stuff. And then I don't know about you guys, but when I drink alcohol, my brain turns off and my nose gets really itchy. So you're gonna see me get dumber and itchier as we go through. And yeah, leafcutter ants. I'm gonna get real confused real quick. So we love your paper. We know that papers are an emotional experience. If you're not a scientist, this is what it looks like for a first pass for science. If you want to see what it looks like for a second pass, Linny and I do our Journal Club podcast where we've read more critically, and you want to see what it looks like to do a third pass, go do a PhD. You just spend 3 years reading the same paper over and over again. Linny and I had a journal club. This is true. Linny and I had a journal club with a woman named Pooja where we read the same paper repeatedly for I think a whole year.
Leading up to qualifying exams.
Right. There's always like for every field, there's like that one paper that you're like, this is my North Star.
Yeah. And yeah, I still don't know if I understand all that paper.
Definitely not. But maybe I did once.
All right, the paper is Neuropeptides Specify and Reprogram Division of Labor in the Leafcutter Ant, Atta cephalotes.
Maybe cephalotes?
Cephalotes. Definitely my pronunciation is wrong. We probably have the graphical abstract. Oh, I wanted to do... Okay, so... Oh, okay. It's going to be... I'm going to change in real time the angle of the camera. It's going to be part of the video. I'm going to do it. I want the camera to be angled down so that there's more of the desk available here.
Okay.
So that when we put the graphics in, they can go here. I'm going to do it.
Like on the desk.
Yeah.
Okay, well, you're repositioning the camera. I also had a bit of a hidden agenda with this paper because I'm trying to appeal to Zach, who has an affinity for ants. So, do you want to tell us about your relationship with ants?
I have a ant colony that I found a queen in our backyard, and I've been raising it for 2 years now.
Wow. So it's in New Jersey.
It's in New Jersey. I found it. It's a local New Jersey ant.
They did not cross
very
state
common.
lines.
Yeah, it's very common. And the first year there was maybe 10 workers, and then this year it got up to about maybe 40. and maybe next year it might be like hundreds.
Oh, these are tiny ant colonies.
Yeah.
Yeah.
So it might get up to, and maybe the 5th year it could get up into the thousands.
So do you feed them, or do they like go
No,
to your
I-
kitchen?
I feed them mostly.
They just leave the
They
ant box.
just, yeah,
They
we
go.
actually just have an ant infestation, and when we call them our ant
And we started naming them. They get chicken. They get, what else do I give them? Mostly chicken. They refuse a lot of things. I try to give them like fruits and stuff, but they aren't interested.
Yeah, we
They
tried.
get sugar water, so I think they get all their sugar from liquid.
Yeah, we tried like blueberries, raspberries, and strawberries, and they only like
They
the
didn't touch 'em.
blueberries. Remember?
Yeah.
Yeah, so they're weirdly picky.
They don't really like dog food.
Do you have videos of these ants?
I have pictures, no videos.
How did you find the queen?
She's just walking around the backyard, and I saw her. So a few years ago, I caught queens. I don't remember how I got interested in it. I think I wanted to raise bees, but didn't have a yard. So I was like, the next best thing is ants.
Great.
And so I found, in grad school, I found a queen ant on the sidewalk. And I was able to raise that one. It had like 10-ish brood, and then it got really hot and I didn't have air conditioning, and the queen died. The workers lived for like another 9 months with no food. And then I finally threw them away.
Okay, so do ants do the same thing as bees, where like if they lose their queen, they try to make another one?
No, they can't,
They
because they
can't,
can't. So, I mean, in the wild, they probably have big enough, and they probably had already started raising another queen, so they would have the capacity to like make a new queen. In captivity, ants, as I understand it, it's very difficult to get them to produce queens. I don't really know how true that is, but I heard that like ants in captivity will not really make queens.
Cool.
You know what we should do for the next journal club? Is the ant active matter paper.
Yeah.
There's a paper from our PhD field of like active matter physics, which is ants.
Yeah.
Oh, that would be fascinating.
David Who, I think,
Uh,
Georgia Tech.
This could just become an ant on
We do kind of like go through these themes of like one paper suggests another paper.
Right, yeah, and there's like a natural progression.
And then all
I
of a
like
sudden
that.
you're an ant expert.
What's up with the correspondence of this paper? The correspondence is the last author, Shelley Berger, and then the second to first author, Karl Glastad. And Karl has given his Gmail address as his contact information. Like on the front page, under correspondence, it's karlglastad@gmail.com.
Well, he must know that he's going to switch institutions.
Okay.
He's a co-first author.
Yeah.
Okay.
Yes.
Okay, I see.
Oh wait.
Is that true?
Yeah, 10 indicates these authors.
Oh, so we
contributed
have 3 co-first authors for this.
Oh my
Yeah,
gosh,
they
three.
use numbers for the co-authoring,
Yeah.
which
Yeah.
is a little bit confusing. But I see. Okay, so that he's the first author contact, and he knows that he's gonna switch email addresses. Okay. So even though Megan has read this paper, because I have no idea what this paper is, I'm gonna ask that we stick to our not great format. Listeners, if you have a better format, let us know.
Well, so since it is cell, should we read the in brief?
Sure. All right, Lenny, hit us with the in brief.
Okay, in brief. We identify specific neuropeptides mediating worker division of labor in the leaf-cutting ant cephalotes, finding 2 neuropeptides associated with characteristic behaviors of leaf cutting and of brood care. This work underscores the essential function of neuropeptides in establishing complex antisocial behavior and remarkable plasticity among individual behavioral types.
Highlights. Characterization of leafcutter ants reveals molecular basis of complex caste system. Diversity of worker types shows neuropeptides defining ant subcastes. Manipulating neuropeptide reprograms ant worker behavior in multiple directions. And comparative transcriptomics shows striking similarity between ant and a eusocial mammal. But which eusocial mammal? Let's find out. Why? I didn't put this and a eusocial mammal in there. They put that in there. Is it just like a really obscure mammal?
Yeah, you're gonna find out
Okay.
in a few figures.
Okay. Next time on Journal Pub. No. Okay. Shall we start reading it?
I want to know who drew the graphical abstract.
I want to know what they used to draw it. I want to know if this was drawn over an extremely long time period in MS Paint. That's my theory. If this was drawn in MS Paint, it actually required a tremendous amount of skill to do.
I feel like sometimes they will like sign it or like give photo credit, but I don't see anything
We
here.
had that where we had somebody, like one of my lab mates' friends, like made one of the figures for us, and then that was like the deal, like that she wanted to go into figure design, I think, for like she was gonna do that as a service. And she signed it, and then the editors told us it couldn't be there.
Wow.
So you had to take out the signature.
We had to remove her signature.
Oh.
Garbage.
Yeah.
Maybe you can put it in the acknowledgements.
I think she made it in the acknowledgements.
Yeah.
We don't want people like.
I don't think the
doing that.
person who made this graphical abstract is going into graphic design.
No, I was wondering if it's a child. No, like a
We
Celeste's
just talked.
son.
We just talked about that people are angry at us for being too critical, and now we're someone in the middle, some middle author spent a tremendous amount of time on this figure.
Okay, I feel like that's not giving enough credit to children. Tremendous
Can we
amount of
appreciate
passion
how
art.
the major ant has a six-pack
Yeah.
and a tattoo on his bicep?
He does have a tattoo on his
Or her
bicep.
bicep.
And then the reprogramming, the same ant still has the same tattoo, and they like have drawn a perspective shift.
Yeah. I'm just saying, there's a lot of detail.
I don't think it's a child. I don't think the child would have done the tattoo and the perspective shift on the
Like
tattoo.
a 14-year-old?
A 14-year-old, sure. A teenager.
Yeah. Like Celeste's son, I could imagine.
I still think it's a graduate student with a sense of humor who did
Yeah,
it
yeah.
as, paint,
It's
as like a
also
challenge.
Yeah.
All
Well,
right.
it was effective. It got to Reddit, so.
Yeah. Yeah, it was effective. All right, so I guess I'll read the intro, the summary, and then we'll start with the intro.
Okay.
All right, the article is Neuropeptides specify and reprogram division of labor in the leafcutter and Atta, you said cephalotes. Okay. The summary, this is different from the in-brief. Social insects offer powerful models to investigate mechanisms of elaborate individual behaviors comprising a cooperative community. Workers of the leafcutter ant genus Atta are extreme samples of behavioral segregation among phenotypically distinct workers. We use this worker system to test the molecular underpinnings of behavioral programming and the extent of plasticity to reprogramming. We identify specific neuropeptides mediating worker division of labor in Atta cephalotes, finding 2 neuropeptides associated with characteristic behaviors of leaf cutting and of brood care. Genetic knockdown or injection of these neuropeptides led to a stark gain of or loss of each behavior and to transcriptomic shifts toward gene pathways expressed in the natural casts. We also revealed global similarities between worker transcriptomes of the eusocial mammal, the naked mole rat, H. glaber. They could have just said naked mole rat on the previous page. That was not a very obscure animal. With orthologous A. cephalotes workers. So we revealed global similarities between worker transcriptomes of the eusocial mammal, the naked mole rat, with orthologous A. cephalotes workers. Huh.
So do we know what eusocial means?
That is like, it means
Is
there's
this
like
Truly
division.
social, truly social. Right? You means truly? Isn't that the
so
core
it's,
of the
the
word?
queen is the only one that can reproduce.
Yeah, or like the division of
It's this
of
like
different
bee ant
tasks.
structure.
Yeah.
So I
Yeah.
think it's.
And the
it's
naked
that-
mole rats have the same
that
structure.
Yeah, I know they had
all-
the same structure. I just didn't.
Most of them don't reproduce. They
Someone
all.
gave a talk on
Yeah.
naked mole rats.
Someone from
Yeah.
the California. It was
It
a
was
great
super
talk.
cool.
Yeah.
Good talk.
Yeah.
Oh man, I wish I remembered her name.
I like
It was an incredible talk. Though.
Yeah.
This work underscores the essential function of neuropeptides in establishing complex social behavior and a remarkable plasticity among individual behavioral types. All right, Linnea, introduction.
Okay, and here plasticity is being used in the neuroscience sense of changeable.
Rather than
Yeah.
unchangeable.
Yeah,
Okay.
which is the soft matter sense. Okay, introduction. An outstanding question in biology is how differential regulation of the genome is translated at the organismal level to regulate and alter behavior during and after development. Social insects are exceptional emerging models to investigate this question, providing a natural system exhibiting striking examples of such plasticity. Indeed, social insect colonies encompass multiple morphometrically and/or behaviorally distinct female types, despite existing as a closely related family with alternative phenotypes that primarily arise from epigenetic, not genetic, differences between members. Individual behaviors are segregated by phenotype, age, colony demography,
Colony demography? I don't know.
or a combination of these and other factors, resulting in a complex reproductive division of labor with typically one queen and multiple sterile workers, commonly referred to as a caste system. When polymorphism is extensive, the physical worker caste may be divided into subcastes according to allometry and trait covariance. For example, worker castes perform the foraging and brood care tasks of the colony, while the reproductively active queen does not forage and mainly lays eggs. In some social insects, including many ant species, tasks are further subdivided among distinct phenotypes of workers. The molecular mechanisms that determine subcaste-specific behaviors in social insects remain understudied, despite recent findings that have begun to identify a complex system of gene and neurotransmitter regulation and activation and maintenance of these behaviors.
One exemplar of worker division of labor is the worker subcaste system of the genus Atta. The complexity of worker subcastes within Atta provides a robust model for investigating mechanisms governing behavior. The intricate subcaste system in Atta facilitates a life history of agricultural lifestyle, representing one of the most complex among ants, or indeed among human society, or outside of human society. In Atta colonies, leaves are collected by foragers, macerated, processed, and then provided as a substrate for cultivation of a fungus that provides nutrition for the entire colony, thus establishing a multifaceted symbiotic system between the ants, two species of fungus, and a bacterium.
Oh no, kill me. I don't think we've been recording this whole time. Okay. Now we're recording. But that thing will catch our audio. So we'll just keep going.
Cool.
Okay. Specifically in the Atta cephalotes, the complex tasks required to manage the colony are divided among workers of multiple distinct size morphologies. The smaller worker subcastes perform multiple distinct specialized tasks within the nest, including leaf processing, management, cleaning, feeding of fungus, nursing of brood, and waste management. In contrast, the largest subcaste morphologies regularly leave the nest and perform duties associated with collecting leaves and territorial defense.
Neurohormones and neuropeptides are critical molecular regulators in establishing and maintaining social behaviors across taxa. For example, in humans, oxytocin is released during caretaking behaviors, such as when babies suckle mothers, to encourage milk release and overall bonding. Similarly, vasopressin in humans promotes pair bonding and social recognition. In eusocial Hymenoptera, including wasps, bees, and fire ants, juvenile hormone, JH, acts to influence social behaviors through its gonadotropin function. Neuropeptides such as corazonin regulate caste-specific phenotypes in ants, including foraging behavior and egg deposition.
Oh, me. Sorry. I'm going to apologize for not recording. So I guess it will be like kind of shitty audio for the first 15 minutes. But did you guys see the previous video? I felt the audio was pretty okay when this thing cut out.
It was okay.
Yeah, I think it would be fine.
Okay. Here we uncovered two neuropeptides and cephalotes that regulate subcaste behavior and where neuropeptide gain or loss of function reprograms behavior. Sorry. First, we identify crustacean cardioactive peptide, CCAP, which is linked to feeding behavior in flies. It is central to association with and movement of leaves, a characteristic behavior of one leafcutter worker subcaste. Second, we identify neuroparsin A, NPA, which is linked to swarming behavior in the desert locust. And why are we going to other animals here? Essential to the regulation of brood care. a focal behavior distinguishing the smaller subcastes from larger subcastes. We extend findings of brood care to the naked mole rat, an exemplar of a mammalian eusocial system, to reveal striking transcriptional correlations with ant forager and nurse worker subcastes. Just forgetting to hit record has really shaken me, and I'm not able to read anymore. We find that NPA engages conserved pathways that are fundamental to behavioral regulation across the animal kingdom. We propose that neuropeptides serve unique functions within the intricate subcaste structure of leafcutter ants to determine complex behavioral repertoires.
Repertoires.
Is that how you spell that?
So I think eusocial is like eukaryotic,
Yeah.
right? Where like you subdivide the
True kernel.
true kernel. What? Okay.
I think that's what eukaryotic means, no?
I think that's right.
Okay. But why is it true to subdivide? Anyways. Are we reading to figure 1?
Yeah, so
moving
you'll
on?
hit us with the one sentence to figure 1.
Behavioral and molecular characterization suggests 4 distinct morphological groups. We built upon prior studies of subcaste in Atta that utilized head width measurements combined with our own behavioral observations to define 4 worker subcastes, figure 1A and S1A.
All right, to the figures.
Oh, but it's annoying
Why is it annoying?
because
the figure caption is far
on
away.
the other side.
Yeah.
Should we share one?
Yeah, I'll fold up mine and I'll look at yours.
Oh, that's a good idea. Yeah, all these figures
What
are
is the
huge.
strategy you're employing? Oh, I see.
So that we can.
Woof, this is a big figure. Okay. Figure 1A. A cephalotes shows transcriptional differentiation between major morphometric subcastes. I think that's a sentence.
Yeah.
Okay.
A plus.
A. Table of primary 4 subcaste phenotypic sizes used here. So they've got a bunch of the ants put together by pins. That seems clear enough. And then they have the subcaste, the head width in millimeters, and the behaviors of the subcaste. And then
As a little table.
as a little table. And then they have namings. Minimum, NMM, minor, MNR, media, MED, and major, MAJ. Cool. I don't know why they bothered to put that, but seems friendly enough. Do they ever use that again in the figure?
I guess it doesn't really look like it.
Okay, sure. Whatever. That's fine.
But yeah, I guess the big ones with big heads are involved in like more defense and things out of the nest.
Now this thing turned off.
Uh-oh, what does that mean?
Alright, so we did A. I think I caught that thing shutting down right when it shut down.
Okay,
So,
yeah.
We'll see. Alright, B. Proportion of time with fungus. What?
So one of the jobs is they, like, feed the fungus, take care of the fungus. That's
Okay.
one of the castes. That's
Sure,
their job. And so they're just looking, and that's the minima caste. Looks like they're responsible for. So the blue on the left spends the most time handling fungus.
What does a data point mean here? A dot?
I think so. I think they show it in B, in the lower part. There's that little box. And so I think they put an ant in this box, and they have access to leaves, brood, and something else, right?
And fungus.
And fungus. And then they basically keep track of how long it tends to the brood, or tends to the fungus, or hangs out with the leaves.
Can
and
I
then
ask?
they also, the big ones, something about biting behavior, so they see how aggressive they are.
Can I ask? Then does each dot mean an ant?
I think they put them in one at a time. Is that right?
Yeah, I think they put. I think they do this with individual ants, and they put each one in for like 20 minutes or
Yeah, I
something
think
like that.
they put them in,
and
they
then
wait
they
5
see
minutes.
how long they're like spending doing each of these things.
But then it looks like N is 5 ants.
Yeah, that could be right.
It seems like not enough ants.
You don't think that's enough ants?
Well, I feel like the first panel to B is fairly unconvincing that the minim population is actually going after fungus more. Like, they can say 1 star if they want, but those 4 graphs look the same to me.
Okay, well, let's look at the other.
Well, I think also if you look at the line in the, like if you look at the majors, they spend 0 time with most of them, right? The halfway line of that box plot is
Sure, but the media
0.
and the minim don't look... I mean, I know this is probably not important, but...
Well, yeah, I think probably there's more overlap with the time with fungus. But then if we take a look at some of the other things like the leaf interactions and the brood moving, we can see
Like
and
strong
and
differences.
in the I guess the biting thing where they're in the middle, I guess maybe
I don't know what they. I didn't
Maybe biting
understand
each
how
other?
they. I didn't understand how they test the. biting.
then it seems like there's more clear associations with a single cast.
So, Zach, did you also read the paper?
I read about 3/4 of the paper.
Okay. So, Linnea, we're just the odd ones out here.
Well, now we get to learn more. I'm
It
pretty
could be that
excited.
the new format is Megan and Zach read the paper and explain the paper to us.
No,
They
I'll
bring
try.
us-
Oh yeah,
They bring us alcohol and explain.
I was saying a possible format to this because like people are like, you know, some people are like, oh, you should be more critical, and other people are like, you're too critical, and people are getting
Sad,
upset.
yeah.
I was like, maybe we should do like one person's job is to attack the paper, and the other person's job is to defend the paper.
Oh,
and
that's
I
a
feel
good
like that takes
idea.
a little bit of the personal thing out of it.
Yeah.
But you guys are automatically on team defense because you've actually read at least some of the
Well,
paper.
because I do think it was understandable when I read it, but then when I just looked at the figure, I'm like, oh, I'm not sure I would have known that that's what this was showing if I didn't actually read that in the text, which is more of just had there been a little diagram, maybe that would have helped. But the data's all there.
The decision to rotate one of the 4 graphs in B 90 degrees is pretty weird. It's fine, but why?
Oh, maybe they were
what occurred that made them want to do that?
But are you ever making a figure and you're like, this heat map will only fit in this corner?
Yeah.
Sure, yes, but I have never, there's some white space here in B. I have never thought, let me take one of these, just one of them, rotated 90 degrees.
Fair.
Okay, that seems fairly clear that the different broods have different...
I find it kind of
Thanks.
whimsical.
I think you're just trying to be nice because people are so angry at us for being
Well,
mean.
you did just mention that too, but also it fits with the
Yeah, I think,
the spatial
I think
organization of the.
they made it the size of D is
Yeah,
what they were trying
yeah.
to do.
But look to the left of that. Look at this white space right here.
But you could have made them all vertical by moving this thing over and then putting this one here.
That's
or you could have
fair.
rotated this thing vertically.
Yeah.
But then the plot wouldn't be right next to the larva.
Yeah. That's what I think is whimsical.
Yeah.
All right,
like
so
that.
now they do a principal component analysis, PC1, PC2, and they put down the things. The 4 guys seem pretty good separation. Why did they say out and in?
So the majors and the media leave the nest, the minima and the minor do not.
Oh, they're saying that PC1 is essentially out and in.
What was this on count? What was this on?
Yeah,
It's on-
so again, one of those things that you wouldn't necessarily know unless you read the paper, but then they do RNA seq on whole brains of individual ants. And this
Of
piece
these ones that they
Wait,
yeah.
measured.
This PC1
wow.
and PC2 is from RNA-seq? I was assuming it was
Yes.
from the data in B for some reason. Okay.
yes, it definitely is, right? I think it it definitely is.
Okay. So these are principal components of RNA-seq.
Yeah, so essentially they're just saying transcriptomically they the genes separate out into you can see the transcriptomes are separated by those that spend time outside versus those that stay inside.
Okay.
Okay, then n equals 5 makes more sense.
Yeah, because then are there 5 of each?
Of each that they send.
Oh, it looks like
No.
there's
like there's
Why-
10.
Why do you bring up n equals 5 again?
Are these not the same ants that they measured the behavior of?
I assume
That is
no.
that is unclear. It could be, but
Oh, you assume no?
I don't know.
You think yes?
I assumed yes.
I think they must do it by size.
I think they might do it by size and behavior to classify them.
Yeah, they use both to. Probably it's the same ants because they would need to have like.
So you think B and C are the same ants? So they did RNA-seq to the ants that they did in B?
I think
And they're
probably.
doing RNA-seq to the 5 ants?
I don't know.
That's my hope, but
Well, why are there 10 dots here?
Yeah, there's a
and 5
lot
dots
of
here?
minor dots that don't.
I'm not convinced that there's 5 dots. I haven't been able to count.
Look on like media of B1. You can see 1, 2, 3, 4, 5 dots.
Okay. But like minimum 1, 2,
Yeah.
3, 4, 5, 6, 7.
No, what are you counting? There's no.
There's one at the very bottom at 0.
Oh, you see there's 2 dots.
And
There
then
might be a
there's
bunch
2
of
dots
zeros.
below
Oh, there's one way
the.
up here. All right,
So-
all right. So maybe it really is the RNA-seq is literally of a dot is an ant.
That would make sense.
Cool. Weird. Cool. Okay, D is this expression RNA-seq disaster.
Figure ruiner.
thing. Kill me. This is just so hard to parse. I don't know, man. It's so hard to parse this kind of data.
But that's okay. I really don't think it's important. I think it's just saying that.
Why is it here?
No, it's just highlighting that there are genes that are specific or
Well,
enriched.
these are pathways.
Have you
These are
ever
different
given a group
pathways,
meeting
yeah.
with this kind of data and put up your data and been like, I really don't think this is important. But
Or it's not that it's not important. It's that
It's very obvious that transcriptomically these ants are very different.
okay, so
So the four.
and the heat map shows
the
this
fourth
very
Okay, so
thing
because
are
the
four
heat
each
map.
of these,
Okay,
Yeah.
okay,
So
okay.
so
each column looks different, is what we're supposed
Yes, and then the big divides I think are probably different types of pathways, but I actually don't know.
Yeah, they focus in on a couple pathways. DSBR via HR, who knows? Olfactory learning seems like an interesting pathway to focus in on. Toll signaling pathway, and
to. Mitochondrial ATP synthesis.
woo!
Yay!
I blew out the thing.
As you should, as
Now I
you should.
favor the paper again.
Why do they focus on mitochondrial ATP synthase coupled electron?
I think it just
transport?
changes a lot, like between the different types,
All right,
right?
who's
Like
got good mitochondria? The media ants have good mitochondria. What do the media ants do?
So the media ants they
to
go
bite,
for it. They go
and they go
forage.
for leaves. Okay,
Yeah,
so
yeah.
Oh.
leaf ants have
They're the
good
foragers,
mitochondria.
so they walk
I guess
a
that makes
lot.
sense. They're
Yeah,
getting a
kinda
lot of
makes sense.
mileage.
Defense ants have mid mitochondria, and the minimum minor, yeah. Okay, cool, great. I'm sold. We can stop reading now. Let's stop. No. E,
E.
regulation. So now what are we doing?
Now we are doing functional terms enriched among genes showing significant differential expression in either direction when comparing in subcastes, minimum and minor, to out subcastes, major and media.
Oh yeah, so these are just the top like 15 Go terms that show up when just looking at genes differentially expressed between the ins and the outs.
Okay.
So just like regardless of like direction or whatever.
And do I remember in the paper they actually like talk about how some of these pathways are like known to be significant in different like other species?
Yeah,
Like,
I
there's
get
like
the
connections between
impression-
these pathways and behaviors in I don't know mammals, but I don't say higher, but other species.
Right, like I feel like they reference a lot of literature from like wasps and like other like eusocial like insects that have been studied, which leads me to believe that there's probably like a large body of literature on just like eusociality in like different contexts, and some of the same like players and pathways show up in multiple different like species.
Okay.
So
Interesting. Do you think that is it known whether that was like, I mean, it must be independently evolved.
I think so.
Yeah.
And they'll talk about it a little bit later with the naked mole rat stuff. Sorry, I don't
Yeah.
want to.
this graph, the negative log 10 parentheses p-value, it just tells you how significant the increase is, not necessarily how big the increase is.
Yeah, you look
Yeah.
at the you'll see that in the heat map.
And
So the
heat
fold
map, you'll
change
see
is-
the magnitude.
Yeah,
Okay.
the fold change, the heat map shows the magnitude, and then they show significance.
And so is that what the heat map directly to the right is supposed to be? Like if I wanted to look at this MT translation that's very significant, how would I look at that somewhere else here?
So that's not actually shown in the heat map. The heat map is only showing those two pathways that are highlighted in red.
Okay.
I see. Okay, yeah.
Wait, is that right?
Probably. Yeah, JH and
Yeah, but those aren't the ones highlighted.
response. Oh, I don't know then.
Oh, because I think okay, so I think the point is in these top 15 Go terms, there's some stuff with hormones, right? And so that seems important. And so then they looked at two different hormone pathways, JH, juvenile hormone, and what's this other one? Ecdysone. That must be another hormone. And they're looking at genes associated with these two different like hormone pathways, and they're showing that those are also very different amongst the castes. So I guess it's more just like hormones seem to be important here.
I think my eyes are gonna hurt at the end of this journal pub.
Just because
Because
this
the text is
is
Yeah.
so small.
You
There's a-
definitely
yeah.
can see that, like, you could
Zoom in on
zoom.
the PDF.
Yeah.
All right, what's F? It's A, cephalotes RNA-seq log 2 outnest over innest versus log 10 base mean.
Yes.
Meaningless. What is this showing?
I like the little histogram on
So
the
this
side.
is an MA plot, which I believe comes from physics. That's what.
Sure.
Yeah.
Why not?
I think it's the M is mass and the A is acceleration. I think maybe that's wrong, and I think we stole it. But basically, expression. So log base mean is expression.
Okay, the bottom is expression.
So you're looking at every gene. You're looking at how much that gene is expressed.
Okay.
And then on the y-axis, you're looking at fold change. So they took the RNA-seq of the outnest, so the two big ants, the two big casts, and
Yeah.
they took the two little casts, and they're looking at fold changes between them. So the taller it is, it means that gene is more highly expressed in ants that leave the nest. The lower it is, that gene is more highly expressed in ants that stay in the nest. The further right, it means that gene is more expressed, like it's just a higher expressed gene. And so often you see that like the higher expressed a gene is, the more capacity you have to detect change. And so you don't need as big of a fold change to say that that's significant. So that's why you kind of get this little curve going on so the black ones are significant and the green and the blue are also significant. This is a MA plot, I think, with multiple
Man, this is a totally
conditions going on, and this is something new to me. Multiple
Well, so
conditions
there's
on one
this
plot, but it's kind of cool.
self-actor and in vitro. What are those conditions?
So
Like-
that's a species. The H self-act, that's a species of ant. So that was one
Okay.
thing I didn't quite follow. They look at different species throughout this. It's not just the one ant, right? The leafcutter.
Oh, yeah, I think it's like so green and blue I think are certain genes because every dot is a gene. So certain like the dots that are green are sets of genes that are associated with juvenile hormone responsiveness in I think RNA-seq from a different
Okay.
species.
So they use.
So I think they
another
pulled
paper.
some other data sets for this.
I see. Okay.
But I think what the take home is that from this set of genes that is considered juvenile hormone responsive in this other, I think this is a carpenter ant species. These juvenile hormone responsive genes that are labeled in green are mostly enriched in the outcasts, whereas these 20E, do you know what that is?
I don't know. I didn't understand.
I think so. I just glanced that they do both in vivo and in vitro cultures, or like they do an in vitro culture of the H cell totor neuronal cells.
Okay.
So they're culturing
I see.
the ant cells and then doing this?
Well, I don't know what H cell totor is.
I think it's another
It's the ant column.
Yeah.
Yeah. And then they do it for the JH3 and the 20E.
those are like those are like pathways, I
Stimulations.
think. So there's a bunch
Yeah.
of genes that are all involved in.
Yeah, so they stimulate these different pathways in culture.
Oh, okay. So they did do this as part of this paper.
This is a vastly different experience than the previous journal pubs, where you guys like know what the fuck is going on.
Well, they say we then compared
Yeah,
this, so it's not clear that they cite a paper. So I think this is from previous literature.
Okay.
Yeah.
But I think the point is that there are a lot of genes that are differentially expressed between the in casts and the out casts, and that these genes seem to be largely similar to juvenile like hormone responsive genes in other contexts.
And the point of the whole figure is just to convince us that these different ants have different transcriptional stuff.
Yes. Yeah.
Exactly. They're just expressing different
So
genes.
they're genetically identical.
They're genetically
clones,
identical.
and yet they have very different RNA or transcriptomics.
Yeah, I was gonna say,
Yeah.
I don't think I need this much evidence, but actually I guess this isn't that much. Like, this is just the amount of evidence, right? You go do RNA-seq, this is the correct amount of evidence. So, okay, sure. Seems great.
Yeah.
Moving on.
It's great to have a full sentence as a figure.
Did this
All we can
paper
ask
have
for.
only
It's gonna be a lot.
full-page figures?
It's gonna be a lot.
I
of
think
heat
so.
maps.
All right.
It's a lot of white space.
So the figure is neuropeptide expression distinguishes morphometric worker casts in A cephalotes, which is a full sentence again.
Okay,
So they're
great.
knocking it out of the park on that.
Yeah.
Shit. I'm too dizzy from the wine already. I can't do this.
Okay, this is a volcano plot.
I know
that,
Okay.
I think. Okay.
So strength, or like how big of a change, is the x-axis. Log fold 2 change. So the further right, the more it's expressed in the out ants, the ants that weave, and the more to the left the gene is, it is expressed in the in ants. And then the y-axis is the significance.
Yeah.
So the higher it is, the more significant. So a lot of things change. But then here they're also looking at a couple. I forget they split out some like.
So a lot of things change, but if we go to B, we're going to focus very specifically on neuropeptides, because not only do a lot of things change, but a lot of neuropeptides change. And that, I guess, kind of checks out. If there are like different behaviors, like maybe neuropeptides would be differentially expressed in the different casts.
And they focus in on specifically the CCAP, the NPA, the SNPF, and the DH31 neuropeptides.
So why do we say neuropeptides and not protein here?
There are proteins, so I feel like you could just say protein.
It's just like a specific kind of protein.
It's just and I guess here
I
these
feel
are the
like
genes that are
peptide
encoding
evokes
for
something
that.
like
Short.
small,
Yeah.
And
Okay.
then
Yeah.
neuro tells you,
that it goes to the brain.
yeah. And so, yeah, it's like a more eloquent,
Yeah, I'm just making sure that it's still amino acids and it's
Yeah.
expressed by a gene. There's
Yeah.
nothing like weird going on.
Yeah, and these
I
are
do
all
know,
the
yeah.
genes that encode for the neuropeptides.
I wonder how many physicists have this. I do have like a really big problem with the word peptide, where it's just like, because it's not the word protein, I think,
Yeah,
for
I
like
had-
maybe 10 years now, every time I see the word peptide, I'm like, huh? Oh, okay.
I had this problem where I was like, can I express a peptide in E. coli? And it's like, I guess I can. It's just amino acids.
Yeah.
But I think there's something that trips physicists up about this word.
I think it's just, it's another word.
Semantics.
Yeah. Okay, sorry.
Yeah, so a lot of neuropeptides are differentially expressed in the different casts, and as Bez alluded, they just highlight like 4 of them that are more or less like cast enriched. So, for example, CCAP is enriched in the media cast, which does most of the like foraging for leaves. Versus NPA is enriched in the major cast, which is like the large like defense one. So, okay, there seems to be neuropeptides that are cast specific.
Cool.
All right, so C is now the RNA-seq log 2 of the major over the minor, or the median over the major expression. I don't know what.
They did it. So they have RNA-seq data and mass spec data now. So they're looking at RNA-seq is measuring RNA, mass spec is measuring the actual protein.
And RNA, when we're measuring, like what we mean by measuring RNA is we mean expression.
We mean RNA expression.
Yeah.
And that doesn't, usually it's correlated with protein expression, but not always, right? There's other regulatory mechanisms that can take place in between.
And so what is literally a dot here?
A gene. Well, a gene or protein, right? Gene on the x-axis, protein on the y-axis.
So
But
via
it's
mass spec, we're identifying the protein that goes with the gene from RNA-seq.
yeah. So you have the amount of RNA that exists in the cell,
This is some fancy
innate
data.
X, and you have the amount of protein of that, the product that was made by that gene.
Lenny, do you have this kind of data? This is
No, I've
some
never
fancy
done
data, right?
RNA-seq, so.
I've never done mass spec.
You guys could team up. Get some data like this. I mean, we do both in my lab. We've never done anything like this.
But it's kind of
Fancy
important,
data.
right? Like if you want to make the claim that
Yeah,
the expression
yeah.
of this gene matters, if it doesn't change the protein expression, it probably doesn't actually matter.
Yeah.
And so
So
here they do make that
We
claim.
always just, at least in the field in RNA, we kind of assume expression is going to lead to actual protein changes.
Yeah.
But that's not necessarily true.
Yeah, I did kind of drill people about this a little bit when I was doing spatial RNA-seq. Of I was like, what are the chances that we have some uptick in this RNA-seq and we do not have an uptick in the actual protein? And there were some skeptics, but the general vibe I got was people are like, nah, it's very unlikely that occurs.
Yeah, and protein degrades slower than RNA probably, so the turnover rate, it can take, you could stop a gene from being expressed, but the protein might be around for a while, and so you're not actually measuring a knockdown of that protein until the turnover of all of the protein is made before you started.
Okay.
Okay, so these plots show us, so the min plot looks very linear, so I'm assuming that means that they're very correlated.
Well, wait, why? Yeah, why?
And
are
then
they
the
doing this?
medium major is
Why aren't they just doing
A little more
RNA-seq
wonky?
of major versus RNA mass spec of major? Why are we doing these ratios between the two?
So is it because the rest of do they mostly focus on major and mediate?
Well,
For the
yeah,
rest of the
I'm
paper?
I'm thinking they want to highlight that
Maybe it's
that the
normalization.
neuropeptide itself, the actual protein, is enriched in one cast versus another. So
Okay, and so it's enriched both protein, it's enriched both RNA.
yeah, because I guess maybe there's an argument that it would be the RNA would be enriched, but the protein wouldn't actually be enriched. But then they highlight MPA and CCAP, which are
I see.
clearly becoming their two that are their favorites.
okay.
And they're showing NPA is enriched in the major cast, whereas CCAP is enriched in the, is it media? Yeah, media cast.
Yeah.
We talked about this, I think, in normal journal club about how comfortable biologists are with white space.
Yeah.
They've got this huge chunk of white space
Yeah.
here. It's a full page figure with incredibly tiny text. And they were like, the white space is fine. We
Yeah,
don't
this
need
is very Martin. He would be okay with this.
Okay, but how much of this is them versus the editor that laid out the figures?
Well, usually the authors lay out the figures.
But couldn't, I guess the editor couldn't have
messed with the figures.
No, but I'm saying couldn't the editor have put a little?
No, they have rules, right?
I guess they gave him
yeah.
a PDF of this
Yeah,
thing,
yeah.
right?
Although this story about the editor saying you can't have your signature in the graphic design still blows my mind. Because I can see why an editor would say that, but outside of that, right? Normal ethics are you get art from someone, you let them sign their art, and I don't see how that ethics doesn't apply to a paper.
Well,
or
how
to,
does copyright law? Yeah,
I know that people
because
That's
they
true, 'cause
then,
then they
own
they
it.
own your signature,
Yeah, the journal owns
and they
the
probably don't
figure.
want to own your signature.
Well, I think it would be they own the
They own the art,
they own the
the-
figure, so
of
they
the
don't want
journal owning
but they
the
would also
figure
own your signature because you would sign it.
is nonsense. The concept of a journal owning your figure is nonsense, and so many people have broken that rule and
I
have
mean,
complained.
yeah,
I'm
yeah.
not going to shout out any people because I'm going to put this online. But I've talked about how people break this rule all the time. And
I mean,
the
it's not a
concept
good
is-
rule, but it is the law, right? And nobody
it's
has
the law
sued on
according
it.
to
Yeah.
the journals. What are they on about? We're educators. The free use thing is incredibly powerful. Science, the non-profit, is going to start suing people for this is nonsense. It's all nonsense.
Wait, because you mean people were reusing their figures that they already published? Is that
Again,
the
not
problem?
shouting anyone out. Someone had a journal cover of a very beautiful picture, which then they also submitted to a I'm trying to obscure it as much as possible. Which they also submitted to an artistic competition, a very prolific artistic competition, and won a very high prize, a money prize for that exact same photo in an artistic competition. And obviously that's not allowed, but also why?
I feel like it should be allowed.
But on the rules of a journal owns the figure, it's not allowed, which shouldn't be a thing. That's a nonsense thing. As educators,
I'm not
creating,
saying it's not nonsense. That's not my argument. I'm just trying to explain why they might not let the signature go in.
Okay, I'm pretty drunk, so we're gonna get pretty. Anyways, we're gonna get sidetracked a few times. Wine, I really like wine. D is a UMAP of.
Oh,
Why do
okay.
I care about this? I mean,
A new species.
Yes, we're a
Oh,
new
this is a different
species.
species.
What species
C.
is this?
This is just an
Floridinus.
ant. I think it's actually very
This
common.
is the carpenter ant, I think,
That would make more sense. The C
because
is
it's the same. Oh no, it's a
They
different
know
one. I don't know.
So much about this paper.
It's great.
I am happy. I just feel guilty. I just want to set the expectation. We will never read papers for Journal Club.
No, because
hate it.
I do think you wouldn't have known what this UMAP was because you didn't read the paper.
It's true.
And then we would have sat here and been like, what is this UMAP?
Yeah.
What are we
They just put so much onus on you guys to read the paper. I
But
feel guilty.
I
And it's
think
a little
it-
bit their fault. Why are they throwing in a UMAP of a different species just with no explanation in the figure?
Well, yeah, so
Well,
I
I think they
do.
explain in the paper.
Yeah. Obviously.
So this is a UMAP from a different species of ant. I'm not sure which one.
I assume it's just they have this data. They don't have single cell data of leafcutter ants.
And it's single cell data, I think, of a whole brain of an ant, and they're looking at the NPA positive cells.
Okay.
So that's that
That's the
neuropeptide.
blue? Yeah.
Yes, that's the blue, and it's very highly enriched in only 22 cells, which is actually super interesting. It's super specific to a little tiny part of the brain.
Interesting.
22 cells.
Yeah.
Yeah, but look how high it is in those
If you had
cells.
to guess, can I assume that these cells are neuronal cells?
Yeah, they're in
Yes.
the brain, right?
If you had to guess, how many neuronal cells do you think an ant has?
Well, this looks like a lot.
Yeah.
Yeah, maybe 500.
More
Oh, wait,
than
do you think the
that.
whole
There's
UMAP?
22. There's
I think
22.
that's a whole
I think
brain.
way more than 500.
I assume that just this cluster at the top with the neuronal cells. Do you think the whole UMAP is neuronal
Yeah, I
cells?
thought she said it was the brain.
It would be
But
pretty
it
easy
might
to
it
at
might
least
still
just be a
have
total head.
it might have oligos and other.
I mean, it was had blood, right?
Yeah, it might have other cell types in it. So it might not all be neurons.
Okay.
But probably the whole cluster is the brain, right?
Probably.
You're gonna take the over or under on 10,000 neurons in an ant.
10,000 under.
You're gonna take the under
On 10,000?
neurons.
That
Other
seems like a
votes?
lot of neurons for
Over
an
or
ant.
under on 10,000 neurons.
Way under.
I don't.
Under.
How many are in a human? A lot. How many are in a
Under?
chicken?
I'm
Maybe
taking
it's
under.
Millions, I assume.
Millions. I've never thought about it. I think more than 10,000.
Wow.
Yeah.
I think also more than 10,000. I'm gonna take the over as well.
How are we gonna find out?
Well, if these are all from the brain, then there's definitely more than.
We'll count it.
There's more than 22.
1.
Percent umi versus a bunch of tiny things I can't possibly see.
Okay, this is just in those 22 cells that were highly expressed MPA. Here's just some other genes that were also really highly enriched in those
Okay.
22 cells.
Okay.
I feel like geneticists and string theorists just have really gone hard on the more uninterpretable they can make their shit, the better. Why is everything so small, geneticists? Please, God, help me.
Well, my question is they clearly want to highlight the red boxed things. So why not make the font on those ones bigger? Like, I understand that all of the other things on the x-axis we don't actually care about.
So actually, okay,
But-
wait. I published a paper. I published 2 papers now, where both times I got yelled at by the editor that my fonts were too small. Right, and they said we have a minimum font size for this journal. I'm sure that Cell has a minimum font size for the journal.
I think you found it.
This is like 2-point font.
It's like 6.
How did they get away with this?
I mean, I guess most of us read our papers on PDFs
Yeah.
these days.
That's
Yeah.
fine. That's fine. It's although
A
I
printed
will
journal
say,
is a crazy thing these days, right? A printed article.
all of my PIs except one. All of my PIs print journals and they print papers and they print them in black and white. So when we talk about this like nightmare scenario of someone's printed it out and they don't have color, really we have PIs. That's how they read papers. So it is a real human being. What am I supposed to learn from this incredibly tiny?
That these other genes are also upregulated in these 22 cells.
What is the
Like-
x-axis? Oh, okay. This
Oh, we
is
don't-
22 cells.
What is the
NPA
y-axis?
cells.
NPA cells. So it's like CG14687 is upregulated. ETHR is upregulated.
But
So
why
if
are
you
there-
knew
what are the
something about
different
these
rows?
genes.
The different
Genes.
genes, yeah.
And then the x-axis?
The different clusters.
Oh, okay.
Yeah.
You're not woozy.
I'm not. I don't feel woozy. No.
I feel so woozy.
You didn't have dinner.
Yeah, I haven't eaten all day, I don't think.
You had some frozen.
Oh no, I had frozen.
Romaine, yeah.
Yeah.
Okay, this one that they highlight and they show the box plot is LLP1, which is also expressed in the NPA high cells, which is like somehow involved in like insulin signaling, but that's all we need to know.
So the thing that we were supposed to get convinced from this figure, which is a little bit trickier, is that neuropeptide expression distinguishes these casts. And do they show here convincingly that only neuropeptides and nothing else distinguishes these casts?
Not yet. That's the next figure. So
Yeah,
they're
so-
showing that they're basically laying the groundwork for why they're going to do the next.
So
experiment.
they're saying that it's.
This is how they
that
found
it
something.
can
Okay.
separate
Okay.
the
But I think you
classes,
have a
but not
point because
that
if
it
you look
has
at
to.
A, there's tons of things that are different between the ins and the outs, and we're only looking at neuropeptides, but like maybe we have rationale to do that.
Yeah, so it's like more like a survey of everything kind of suggests to
yeah.
us that we should be looking at
I think that may be why they're
neuropeptides.
pulling in other species because like they don't have data in their species. It doesn't
okay.
exist, and so they're showing in these other ants, this has been linked to something, and we're going to take advantage of that to move our make a decision.
Okay. Okay. Yes, this time we get the figure on the same page as the paper.
But we don't get a full sentence.
Oh, behavioral changes
or
with
the-
NPA and CCAP, not a full sentence.
The legend does
Boo.
also
You
spill
could.
onto
It
the
would be
next
so
page.
close too. You could just say behavior changes.
Fair.
We do get another figure that looks like it was made in MS Paint.
Oh, with the drawings.
Yeah.
Yeah.
I do like these little ants.
They're cute.
Yeah.
I feel like there's someone out there screaming who like is a graphic designer who has spent 100 hours making some figure with Adobe Illustrator, and they're like, I could have gotten more attention if I had just spent an hour making figures with MS Paint.
I think that it's really something is really endearing about hand drawn, kind of shitty.
But this is interesting, right? This is not hand, right? Like, can I draw on a piece of paper my figure and upload it to Cell? Like, would that also be endearing, or is it only endearing because it's MS Paint?
I think it's also endearing if you were to draw
If I
it.
hand draw, because I would love to. I would really love to hand draw figures and upload my hand drawn figures.
You're gonna be in for a real treat when you see
Oh,
figure
is that a hand-drawn
figure
figure
6 or 7.
Insane. Okay.
I guess we have to move along then if there's 6 or 7 figures.
Yeah. I guess how long have we been recording?
I think a long time.
Let's see, I can go see over here. We have been recording for an hour, so we do have to pick up the pace. All right. Behavioral changes with NPA and CCAP. The brood moved What are we learning? Brood moved. Minor moves. Nothing
Okay,
else
so
moves.
they're moving brood around. So that's a behavior
Oh,
of a minor
I see,
of
yeah,
a
yeah.
minor
Okay,
cast.
okay,
They
sure,
move.
sure.
They take care of brood,
Yeah.
Okay.
So then they do a knockdown in major.
and they knock down which one? This is NP.
NPA,
NPA
yeah.
at the top. So the one of the ones they called out in the last figure.
Okay.
So they took major ants that in A never care for brood. They knock down NPA or VKR, which was in the same pathway. I don't remember.
Yeah.
Above or below.
No, so below
And then
they
it
have a little diagram. They
Yeah.
show that MPA is the neuropeptide and VKR is the receptor.
Okay. And then the scramble is the GFP. So they, or not scramble, but they just knock out a protein that doesn't exist, and they behave like normal majors. And so by knocking down this gene, or
They increase nursing.
they increase nursing. Yeah, so they can basically make the ants behave like another caste with one protein or one gene knockdown.
Isn't
two
that
gene
cool?
knockdowns.
Yeah.
It is cool. The
It's
whole
so
thing is
cool.
Yeah.
I'm like super
A
into-
single they knock down a single gene and it like converts casts.
Yeah.
I'm not. Yeah, I mean, ants are great. I'm like, I've been talking about this kind of stuff for bees for a long time. Like the whole thing is super cool. Just like reminds me of my bee.
And so presumably, if you knock down this one NPA, then you're getting all of this change.
I think that's
they
yeah.
discuss that in one of the figures, right?
Okay.
They start showing. I think they go into RNA-seq
I think that's the
of
assumption.
the knockdown,
Yeah.
and they show that you're making huge changes. As well.
Yeah.
Should we?
It's at like the top
Maybe
of a
it doesn't finish
pathway.
the line, though.
I'm working on it. But you guys can get more.
I think we'll wait until you finish and then we'll pause.
We do have to make it at least through figure 3 before we
Yeah.
take intermission.
Yeah. Okay, so we do that. We do a knockdown in media. We do peptide injection in minor and knockdown in C. floridinus, which I still was.
That's another
different
ant species.
Yeah. So then with this next layer of information, they have nursing down, nursing up, nursing up, nursing up, nursing down. Okay, that seems reasonable.
But so they do it both the knockdown. They knock down NPA and they inject it.
Yeah, so the
So-
injection is showing that it.
How do you think you do a injection into an ant?
They said they just.
It said
injected-
just injected in
yeah,
the brain.
That's what they described it. So they. I mean, you can do it flies. You can inject fruit flies. They have machines that I don't know what they look like. Presumably, just like a little needle that you touch to them, and it can inject a very precise amount of.
Sure, I can
food.
imagine doing an injection into a fruit fly. That's within my repertoire.
Okay,
the
but
fruit
it-
fly doesn't move during that.
But a fruit fly is smaller than an ant.
Well, the ant might be asleep.
Yeah, they might. They
Think
might.
put the ant to sleep somehow.
Maybe.
Yeah, like
Yeah.
fruit flies, they put a little CO2
Yeah,
thing.
that's what I'm wondering.
So maybe
Is it
they
the
do
same
that with
thing for
ants. Maybe.
All right, fine. This seems possible. Sure. I can imagine doing that. Okay, so what is then this MS Paint layer?
That's just the diagram
That's
of
just
the
ants.
little ants.
That's the different kinds of ants.
What do you mean
So
that's
in-
just the diagram?
So
Is
in
that
a
no
minor
information?
ant, this is from a minor ant's brain. This is what's happening. In a scramble, you still are able to nurse, but in the NPA injection, you're not able to nurse.
There's no spatial, because at the bottom, it's also peptide injections in media. You got to read this
Yeah,
figure.
again, they're
for
about the
top
whimsy, right?
And bottom. But how do you talk about this in the text?
Well,
See,
the
Are
bottom,
they
we're-
like C1A and F at the same time?
No, they talk about.
the bottom is all CCAP. The top is all NPA.
So we're zooming in on ant brains. That's
Yeah.
what we're
Kinda are-
showing.
I'm just flipping to the text for a second. They're kind of like figure 2B, figure 1B, figure 3A, figure 3C, figure 1B, figure 3D. They're really like all over the place.
I love it.
This is insanity. This is insane.
No need to be linear about your science.
I do think that is a rule, right, with publishing that you
spent
have to.
You do have to,
Which
but it's
I don't-
so annoying.
I don't
Making
think
sure
that should be
everything
a-
is
I don't think
linear.
it should be
That's
a
not
rule.
important.
Yeah.
Like
It's
they
not-
have
and
all the
It's
figures.
never how you
I
actually did the
Feel like I will
experiment,
go
right?
to science jail if everything is not strictly linear.
I just think that like if especially for these kinds of things where if somebody is just reading the figures, like it will go in a different order than somebody reading the paper,
That's
and
true.
that should be okay.
That's true.
Yeah. Yeah.
I don't think maybe when
No, I mean like
I don't think I'm convincing any of my co-authors that we can do whatever we want for linearity. There must have been some interesting conversations about.
Okay, but let's focus.
All right, but that's the whole figure. So
Okay,
the bottom is the same as the top.
So the bottom
no,
they
no, it's
the
a
opposite.
different.
Yeah.
It's the same.
in the bottom
concept.
they inject CCAP, and then the majors start collecting leaves. So it's like their
Okay.
graphical abstract. You have like the buff majors doing media tasks.
Do they talk about how long it takes to go between these two states?
I think they said they test it
figure-
48 hours after. Or
48
something.
hours.
Yeah,
Wow.
Yeah, I remember it. I think it was
Wow,
super
that's
quick.
so fast.
This
Yeah.
figure does not need to be a whole page.
Okay, but look at the difference of the minor head to the major head.
That does not need to be here.
Well, they don't. I mean.
No, but I'm saying
Oh, yeah.
for 48 hours for the minor to then become a major just
No,
because of
it doesn't become-
CCAP?
It
They
doesn't
don't
become
physically
a
become-
size.
Yeah.
It just becomes a behavioral.
Yeah, they only.
It's not literally like this is not the Incredible Hulk that it's just like. Right?
Oh, I see. Okay, that's fair.
And the major doesn't shrink.
This figure could have been a normal size figure. The other one, sure, they had to take up a whole page. This could have taken up a half a page.
Okay, but this is arguably the most interesting figure.
But it did
And
not
therefore
need...
should be the largest, don't you think?
Okay. 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 All right. Do we have 7 figures? Do we want to take a second to form more lines?
Yeah, let's take a second.
Take a break.
We have 6 figures.
Okay. NPA manipulation. Figure 4. NPA manipulation. Sorry. It was really helpful for the editing. We did this very, very cheesy, like, figure
Yeah.
4! Extremely useful for editing this together.
I thought that I thought about doing it earlier, but you had said that you could just see it on the papers.
Why does your figure
Oh,
look
'cause
different than my figure 4?
You're on figure 5.
I've skipped a figure.
NPA manipulation illustrates centrality of NPA for major worker expression. Is a sentence. I don't know what that sentence means, but it is a sentence. Centrality.
So
of
centrality.
NPA.
I think it's because I don't understand what centrality means here.
Yeah, that is a weird.
NPA is important for major worker expression.
So
So
it's
by
a
manipu-
central factor in.
By manipulating NPA, we see that NPA is important.
All right. Subpanel A, we have PC1 and PC2 for actual RNA-seq, presumably. Except for it's been broken into like a really weird... See, all of a sudden we have a lot of graphic design. I feel like this figure has been made genuinely by a different person than made the previous figures.
Well, there are 3 first authors, so.
Yeah.
They split up the figures.
And so now we have someone who cares about white space. And like the font got bigger and everything.
This is nice.
It's luxurious. But what am I
It's
supposed
just
to
there's
learn?
no heat.
Okay,
maps.
so
that's
they
true.
did RNA-seq in the NPA, RNAi, and injected animals, ants.
And bases.
Okay, they did RNAi
Yeah.
to mess with
NPA.
So they knocked down NPA,
Okay.
And they and then in
which should
that's
make,
AB is knockdown and CD is the injection.
Okay.
So knockdown should make, if we go back, it should make the majors into minors.
Yeah, and here for the baseline, the majors are in green, the minors are in blue. And then if we look at the RNAi of a major, it's shifted from like the green side of the graph towards the blue side of the graph.
Okay. But only in the x-axis.
But that's the principal component 1, so that like makes up most of the
Okay.
variation.
Okay. Okay.
And then same thing for C. If you inject NPA into a minor, what you see is that it shifts from the right side of the graph in blue to the left side of the graph in green. So it makes the minors more like majors.
More like majors.
Transcriptomically.
Yeah, okay, cool.
Yeah, so not just behaviorally, but transcriptomically.
Cool.
I might have even considered reversing that in the paper because, like, if you first show that it's transcriptomically it's different, and then show that actually affects its behavior.
Yeah.
Might be interesting.
What are the circle plots in B and D?
Okay, so my understanding is that this MA plot is the same one that we were looking at in the previous figures from like the baseline RNA-seq, just showing all the differentially expressed genes between majors and minors. Or it might not be the same exact plot, but it's showing
gray.
yeah.
In the gray and black.
Instead of the ins versus outs, it's majors versus minors.
Okay,
so minors they skipped the media, so there's a bigger change.
plot.
Yeah, no, I guess it's not. So yeah, this is just showing all the differentially expressed genes between majors and minors. Like regardless, there's no like manipulation here. This is just like the baseline.
But what are the circles?
Okay, so the circles are the green circle is all the genes that are enriched in majors. The blue circle is all the genes that are enriched in minors. The light blue circle is all the genes that are downregulated upon MPA RNAi, and the red circle is all the genes that are upregulated upon MPA. Did I get that right?
I think you did.
Yeah. I like this.
So I think it's kind of cool. I've never seen like a MA or volcano plot with a second dimension on it, and I like it. It's a little bit harder to get your head around at first, but I think it's kind of cool.
So wait, there's overlap for the NPA knockdown between
Yeah.
the upregulated and the downregulated.
Well, so this gives
What
you.
does that central area mean?
I mean, there's some noise in the data, right?
But
Like you mean?
how can a gene be both upregulated and downregulated?
Well,
it's after you do the knockdown. So that's the problem is that if you would have to show like 4 graphs or 2 graphs.
Is there 2 experiments going
It's
on here?
upregulated in one and downregulated in the other. So it's the grays and the blacks, I think, are like just the difference between minors and majors, and then the blues and the reds are once you do knockdowns.
Okay.
Once you do the knockdown.
I
of
see.
NPA.
I see. Okay.
So it's possible.
And then
that a
this is
gene
just like the ones that are totally overlapped are genes, right? It's not that they're enriched or not. Yeah. Genes that exist in all these different data sets.
Okay, and then for E, CCAP subcast X treatment interaction effect. Media versus minor baseline. Log 2 of media over minor.
So, but there are all of these things that are
I don't know, you guys are really
upregulated or
enamored
down.
with these plots. I have a lot of difficulty thinking
Well, but like
critically about these plots.
in the Venn diagram, there's all these things that overlap with the major or the minor, but then there are a bunch of stuff that doesn't.
Right. So it's almost like the knockdown changes a lot of things.
Yeah.
Some of which are associated with like the major versus minor phenotype, but some
Yeah.
of which are just maybe like
Changed
doing
for some
other
other
things.
reason.
Yeah, I guess so. But so I guess Bez, the takeaway would just be that when you like transcriptomically, when you knock down NPA, which is this like major associated neuropeptide, the genes that get down-regulated are associated with a major cast
I think they're
phenotype.
just showing that neuropeptide is causing
Yeah.
like the different behaviors.
You would say it's central
Yeah.
to the major phenotype. Okay, and then the interaction effect, to my understanding, was just like, I think now we're just looking at the genes that are up or like are down regulated upon MPA RNAi and are up regulated upon MPA injection.
Mm-hmm.
So it's like an interaction effect, if I'm understanding that correctly.
I didn't get this far.
But I think it's like the genes that behave exactly how like you would expect that they're both going up when you inject it and they're going down when you inhibit it, and vice versa.
Okay, so this is like genes that they think are more likely to be important.
That's my understanding.
Okay.
Okay.
Next figure.
So it's central.
Okay, back to a full page figure. Weight space. I will say, right. Oh, no, that makes sense. Okay, yeah. NMR shows correspondence in subcaste bias with acephalotes across 600 million years of evolution. What do they mean by NMR?
I don't know. What do they mean by NMR?
Is it MNR?
MNMR.
That's the minor, right? They said that in the beginning. Remember in the beginning when we said that? Why did they use these three-letter terms?
No, it's MNR for the minor. This is NMR.
Oh,
Like,
So, is this not nuclear magnetic
yeah,
resonance?
that's what I would have guessed, but
yeah, but it doesn't look like
I don't
nuclear
think so.
magnetic resonance data. So
there it must be a.
Wait, do you think it's NPA?
Do you think it's
NMR shows correspondence.
NMR. It seems like too big to be a typo, so I feel like they actually meant NMR.
Okay, what else could NMR stand for in this context?
Okay, what figure number is this? Figure?
This is figure
Figure 5, right?
5.
It reads like a technique, right? NMR shows correspondence in subcaste bias with acephalotes across 600 million years of evolution.
So, do you think it's like,
This is what we said we wouldn't do, is just sit here and
Yeah.
try to actually figure something out.
But what is NMR? I don't, like, skimming through, I don't see NMR anywhere in the text. And as Megan has actually read the text.
and I don't know what it is.
Okay. NMR shows correspondence in subcaste bias with acephalotes across 600 million years of evolution. Let's just go through. Let's
Yeah.
see what it says.
Seems legit.
We don't know what NMR is. We'll figure it out.
Okay, yeah.
Okay, so now
A,
The-
cephalotes, and H, glaber. Is H. glaber the naked mole rat?
yeah, now
Okay.
we're introducing a new species, naked mole rat.
All right, there it is. It's beautiful. And then we have M and
Okay, so
M.
apparently naked mole rats also
MNR.
nurse and forage.
Naked mole rat.
Naked
That's
mole
what it
rat.
stands for.
Oh, naked mole rat shows correspondence in subcaste bias.
How did we not get that earlier?
All right. I feel like there was enough room to type it out.
I feel like the overlap between naked mole rat and nuclear
This is probably one of those
resonance
literally like the
is,
figure is only allowed to have 30 words. And
Yeah.
That's what
They were like, okay, naked mole rat, MR. Oh my gosh,
Should we
that's
start
hilarious.
calling naked mole rats MRIs?
Do you talk about them a lot?
Every time you talk about
I
NMR,
would say that Linnea brings up a naked mole rat maybe once a month.
I do.
I would say that you're rather enamored with the model as a research organism.
I think they're a cool model. But I was talking to a postdoc who actually has to work with them, and they work, or like they live in like 90 degrees Fahrenheit and
Oh.
are really not robust to any temperature changes. So you have to go in and like you're just sweating,
Oh, no.
because it's
That's what
very
you have to do
uncomfortable.
with the anoles as well. is
Yeah.
that They're just like high humidity, high temperature creatures.
oh, the humidity would get
Yeah.
me.
And so like keeping a colony is very high maintenance.
Yeah.
And yeah, they're trying to do it in New York City, which seems challenging.
Not the best place to
Yeah.
raise NMRs.
All right, so what am I supposed to see in the MRI feature?
Okay, so they
Nurse versus
did
forager.
RNA-seq on
I guess what is
Not
There's a
what is
difference.
the, what is the top and the
Hypothalamus.
bottom here?
Well, so they've just somehow sorted the genes.
Yeah,
And
my
they put them in the
question.
same order.
And they put them in
Okay.
the same order in the ants and
they,
the mole
They
rats.
clustered the ants.
They clustered
They
the
clustered
ants.
the ants. That's why like the
Yeah.
blues and the reds separate. And then they put the naked mole rats in the same order, and you still see
Some clustering.
the clustering.
Cool.
So like it's similar.
So they did RNA-seq on the hippocampus of
That's-
nursing
That's
versus
so
foraging.
wild,
Super
though.
weird.
So, like, when did these
I'm
guys
not happy about
diverge?
Like I think it said like 600 million
That's
years.
so crazy.
What is the hypothesis here? The
Yeah.
hypothesis is that like all social creatures have like conserved a thing.
Or they convergently evolved the same.
But this would suggest not
Yeah,
convergently
yeah.
evolved. This would suggest conserved.
Well, it's convergent. They convergently evolved the same trait, as in they gained the same trait independently of one another.
You interpret this as they did this system independently rather
Yes.
than they managed to keep a system. Or,
Yes,
like,
because
return on a system.
you're assuming that the common ancestor of flies and naked mole rats were eusocial.
I am not.
Yeah.
That is
I'm
what you're
deeply
assuming.
not assuming.
That's
No,
if you're
what I am
assuming it's not
assuming-
convergent. That's what you're assuming.
What I'm assuming is that there was a set of genetics that wasn't being used in the common ancestor, but could be turned on later by both of them.
Yeah, that's
Yeah, but that
somewhat.
seems still kind of convergent-ish.
Yeah, it's a
I
somewhat.
would call that conserved. If you're using the same material,
But to do... Okay.
It's a somewhat like abstract concept, which I would refer to as like deep homology, which is
Okay.
that like there's only a few ways to do a thing.
all right, this thing is red. So it would like
Can you
that to
can you
end.
plug it in?
Oh, can I plug it in? Okay, I'll go try. Keep talking. Go through the figure. I'll try.
Okay, so this
okay,
is really
so deep
interesting.
homology.
Deep
So
homology.
it just means that they have started with a lot of the same parts and they
Right.
have ended with the same.
Yeah, or like if you only have
Organization.
like a set amount of like genes and like mechanisms by which to do something, you're kind of limited by that, and therefore like very different like lineages will utilize those same like parts to do like similar functions
Okay.
in like different contexts.
Wait, sorry. How are they picking these genes?
I think these are the genes that are just enriched versus in versus out.
Because wouldn't you also just expect if you just took
Yeah, why
a
would you
random set
expect
of genes, but if you
overlap?
just took a random set of genes between yeast and humans, like
there'd be no
oh
homology?
no, a lot of them do the same function, right? The gene does the same thing. So like our RNA genes are highly expressed across all species. Is that not what this is saying?
Well, but we're saying that they're differentially expressed between
they're
nurse
between the
and
nurse
forager.
Okay, that's different. Okay, so the behavior is linked to the
Yeah, so I think they were saying like the young
That's
young
important.
rats nurse the young, and then the old rats go and find food.
which is what bees do,
Oh, okay, so it's like
Right.
age based,
right? Isn't that how bees work? They young bee, because it's different between ants and bees. I think young bees do like stuff inside the nest. As they age, they change and they go do stuff outside the nest. So a single bee goes through phases of their life where their entire body like reprograms their transcriptomics change as they go through life.
I think that's correct.
Ants-
That nursing bees, I think nursing bees tend to be younger, and sometimes they don't have flight capabilities yet.
And
God,
then
I really hope I'm not getting
but-
that wrong. I'm
but
really trying.
I believe they transition into another type as they age.
Yeah, so then they like get better at flying, and then
And then they leave. Ants
they leave.
ants are not like that. Ants
but I will
you
say-
are born into a caste and you do that job. They physically get bigger, the majors, or they're smaller, and they don't throughout their life change their role. They have
These are
a
pretty
role.
weird, right? Like the casts, I don't know, right? Like the main casts are your drones, your worker bees, and your queen, and those casts are sexual. That the drones have like they will meet with the queens.
Yeah, the males.
Yeah, and then the worker bees will not mate with queens, although they can lay eggs in an emergency. And then there's the queen, of course. So this concept of caste of like you are a nurse bee versus you are a like forager bee. I don't know if that comes with a real
No,
phenotype.
because I think they go through. I think the
So
I
these
believe the
subcastes are like
transcriptomics
an age
of the
thing.
different like they take on different roles as they age.
Uh-uh.
And transcriptomically, like they go through a phase of like inside. I take care of the nest. I clean.
Yeah.
I make honey
Yeah.
or whatever they do.
But I guess my question is that a behavior? So you're saying there's a transcriptomic change? My question is that is a behavioral change, or is there also a
It leads
phenotype
to
change?
it. Yeah, as they age, the transcriptomics. I think they're all. There's no like bees that are bigger than other bees. Like,
Yeah, can you? I don't think I can tell the difference between nursery bees and
No.
and
I believe.
But also my question, like, when in winter time,
Yeah, this has been my question for years.
that
yeah.
all the workers are inside bees,
Mhm.
that whose job is to flap. So like, are there
I assume like, as
transcriptomes
it gets colder, they all shift to stay
to
like
stay?
I'm gonna stay inside.
Yeah. And there's some
I
signaling.
mean, ants
happening.
hibernate too, so they
Yeah.
like kind of as things get cold, they change their
Yeah.
behaviors.
Okay.
Do you think this is gonna be the worst video we ever make?
No. I think there's worse yet to come.
I think it's better than our first one, don't you think?
Yeah.
Oh, I hope so. I think that first video was so bad that the last author on that paper won't talk to me anymore. I think he's like, man, these people are morons. Okay. What am I supposed to learn for the rest of this figure?
Okay, yeah, we
have
got a lot of MA
more-
plots
I should have
still.
paid so much more attention to Zach when he was describing how these figures work. I did not realize that these figures were going to be every other, or essentially every figure. Yeah.
Okay.
I mean, essentially, okay, so B, all it's showing is genes that are differentially expressed between nurse and forager types of naked mole rats. And then we're overlapping the media enriched genes and the minor enriched genes. And we can see that those are also like the media enriched genes are forager enriched and the minor enriched genes are nurse enriched. So it's kind of like showing in a different way the same thing that was shown in A, which is just that many of the genes that are defining the different castes in ants are shared amongst naked mole rat
Okay,
castes.
here's my problem. Right, like I was fine with these graphs when the statement seemed obviously true. But this statement is a like amazing, incredible statement. And to make an amazing, incredible statement, I really want amazing, incredible evidence. These graphs don't, like, the data might be here. I don't know. This is a big statement they're making. It seems extremely unintuitive.
Because maybe, like, I mean, I guess maybe we don't know exactly what these genes are. So it looks like on the far right, they have some Go terms associated with it. So, like, maybe there's an argument that, you know, having a forager phenotype is associated with some, like...
This is the problem with genetics. Don't give me all 20,000 data points at the same time. It's ludicrous. I can't think about this critically.
So I do think that is one, I would say, criticism of this paper is it's very much like we're not really getting down, aside from the 2 neuropeptides that we're testing, we're not really getting down to brass tacks as to like what genes are differentially expressed.
So for the 2 neuropeptides that they test,
Those were
do
enough
they
to
do
change the behavior.
MRI versions of those neuropeptides?
So, okay, well, let's continue.
Is that the next figure?
No, but okay, so in C, do you want to go through C?
I didn't read this. I,
Oh, yeah,
I
but-
can't
NPA,
explain.
RNAi, and GFP. So this is NPA. This is the one we
so they
care
are
about.
doing, well, okay, so they're doing NPA RNI into the naked mole rats?
into the ants again.
No, so yeah, again,
Then
here
why,
they're...
so why access the naked mole rats?
So
No, this
here,
is the forager
no,
versus
so
the nurse
so
naked mole
here
rats.
they're showing, like, you know how in B they showed that the media enriched genes were enriched in the foragers. Now in C they're showing that the genes that were upregulated upon
What?
NPA RNAi are enriched in the nurse
Naked mole rats.
naked mole rat.
So the genes that are
Cast.
downregulated
Oh, Okay.
Yeah, so the same genes are changed.
Okay.
In-
But they are not doing an NPA RNAi onto the naked mole rat.
No.
which would be
No.
ludicrous, right?
I mean, that would be awesome, but
I don't think. Anyways.
Yeah,
you don't think it's possible?
Yeah, I don't
because
know. If,
NPA isn't really there.
Can you do this RNAi trick on like full blown freaking mammals?
I mean, I guess
know,
in theory you can
maybe not
inject
in
an
into
RNAi
their brain.
into the brain.
Did that work?
So I do
Did that
know.
work on
How would it
birds?
get everywhere? Yeah.
So I do know, like, the people at like the Neuroscience Institute, they'll like put like viruses into the brain that they actually inject that encode. I don't know if they encode RNAis, but like they put viruses in the brain that will like overexpress things.
But those are viruses, right?
Yes.
Like that I can
Inherent-
see.
those are
Yes,
inherently
yeah.
RNAi
So-
I don't really see.
Yeah, it just like can't get everywhere. Is the like it's a spatial
Yeah,
problem. Yeah.
Okay.
So what they do is they have, and let me check
NPA
the.
stimulation is what they do for the mole rats.
But what does that mean? What does it mean that they do NPA stimulation?
Um,
See, now I'm not like trying it all. I'm just like, Megan, explain this to me.
Okay, so I guess it doesn't even really say in the legend, but here, to my understanding, they have in vitro cultures of astrocytes from naked mole rats.
Okay.
Do you want to switch? I can go look at
Wait.
the methods.
No, I want to look at the text actually. So they don't do any, I don't think they are able to do anything in vivo, but I think they have some cell culture stuff.
All right, I'm reading some of the text because I think this is important. Having established the efficacy of in vitro NPA stimulation using ant cells, we then treated H. glaber astrocyte cultures derived from brain cortex of newborn pups with NPA and compared in vitro NPA stimulation DEGs, to
Differential
H.
express genes.
differential express genes, H. glaber forager nurse differential express genes. Strikingly, genes upregulated by NPA in H. glaber culture were enriched among H. glaber foragers, whereas genes downregulated by NPA in H. glaber culture were enriched among H. glaber nurses. Therefore, NPA repressed genes associated with nursing in H. glaber via potentially conserved. Therefore, NPA repressed genes associated with nursing in H. glaber via potentially conserved mechanism. It's not a sentence. Why does it end with a period? Therefore, NPA repressed genes associated with nursing in H. glaber via a potentially conserved mechanism. Period.
NPA repressed genes.
Oh,
It-
repressed is the verb.
Yeah.
Okay. Therefore, NPA repressed genes associated with nursing in H. glaber via potential conserved mechanism. Fucking kill
I
me.
feel like this is the peak of journal pub that we had initially envisioned.
Yeah, we didn't quite get there during the first few, but
Cool. I seem to remember there being something about is MRI culturing, cell culturing a new thing? I seem to remember something about this talk that this has not always been around as an available technique.
That cell culture in the naked mole rat is kind of new.
Yeah.
I believe that's true that they've had to. Yeah.
okay, but
That
the
is a
cool
recent-
But
thing.
here paste
But
dividends.
they just
Alright.
don't get cancer, right? That's why they're very interesting.
They also
And they're
live super
reproductive. Yeah.
Oh,
They
I
live
think for a
super
long time.
Yeah.
I
The
think-
queens specifically live a
Yeah, they
super
don't go
long
through
time.
menopause for a really long time.
Yeah.
And like an interesting thing is like the
long
queens.
meaning
Like you can be
for
like
for a rodent. That's
you can be like an 80-year-old and have a baby.
Oh, okay. So they reproductively live. They're reproductive a long time.
But I think they also
And
But do
they're-
they
live.
I think they live a
Yeah,
long
they
time
live
too.
a
Because
long
rodents
time.
live like what, 2 years on?
Yeah, so I
No,
think,
but the
I
naked
think
mole
naked
rats
mole rats
live-
are like an outlier on like the
So
how
they
long
live
do
for
you live
like
thing.
30 years.
I
Do they
don't know
outlive
the
humans?
numbers. And then the
Oh.
queens live significantly longer than the non-reproductive members, and the queens also live much further underground. So I think it's thought that there's something different about their metabolism because the respiration is much more difficult in their environment than the rest of the naked mole rat environment. We should do that paper. Screw these ants. No, I like the ants. There's a lot of places we could go from this, right? There's the active matter ant paper that me
Yeah.
and Lydia know that I don't think we've ever read. It's like a really big meme in our old.
I think it was this
PhD.
person, Diana Laird.
Oh, that sounds
It
right.
does look right.
Yeah.
Yeah.
Uh,
We could do Diana
UCSF.
Laird's papers
Yeah.
on naked mole rats.
We'll be in a eusocial animal podcast.
Kick
Yeah.
for a while.
Weird kick. Okay.
Okay, but the point is, I know it's not in vivo, but the point is, if you just in vitro, if you treat these naked mole rat astrocytes with NPA, which doesn't exist in naked mole rats, for the record, it has a transcriptional response that shifts it from.
Wait, NPA doesn't exist
What
in
are you
naked
fucking
mole
talking
rat?
about?
No, that's what it says. So
The receptor's still there.
What do you mean you treated them with NPA then?
you just put
You just,
NPA
like, take
on
the-
the
Put in
cells.
a protein that they
Yeah.
don't have.
Yeah.
And then they begin to behave in a manner.
They respond.
Yeah. Yeah, it
Cool.
says that is despite being absent from H. Gilbert, the naked mole rat, NPA regulates an overlapping set of conserved genes associated with caretaking in both species.
Okay, so here's my problem, right? This is super cool. This is amazing. I want the entire paper to be that one experiment.
Right.
Because that's the amount of evidence that I need to believe something that batshit crazy.
Well, I guess the argument is that even though this peptide doesn't exist in naked mole rat, there must be an orthologous method mechanism by which this is happening in naked mole rat.
Yeah.
Maybe using a very similar protein.
Same receptor.
Yeah, they talk about in the supplement there's some stuff on insulin signaling and how that actually might be the orthologous mechanism.
Fine. This is fine. This is a survey paper. This is all good. You give a Nobel Prize to the people who do this research. Nobel Prize should go to the person who wrote an entire paper about this one experiment rather than the survey paper. That, if this is true, proving that this is true is the single most important thing.
Okay, so you think if they injected NPA in, say they overexpress NPA in the brain of a nursing naked mole rat and it increased its foraging behavior.
Yeah.
That's the evidence you want.
Yeah, and I want it 5 ways, yeah.
Okay.
Because it's a big
Yeah.
claim, right?
Yeah.
I think that is approaching major scientific breakthrough territory. That you get that claim for real, you've changed genetics and neuroscience appreciably.
Right. Yeah, and I would say that the most exciting part of this paper is in ants when they do this, they can actually change the behaviors such that they exhibit behaviors of another caste.
Yeah.
And this whole kind of cross species thing is just a cherry on top that it's kind of it appears across many very divergent eusocial animals
But
that
it's
actually the mechanisms by which they're doing this are probably pretty conserved.
Big cherry on top, right?
But they wouldn't have even had to have naked mole rat data in this paper. And it still would have been a great paper.
But I don't think it would have gone to Cell.
I mean, I don't know about that, but...
It wouldn't have gone to Cell. Well, I don't know about that, actually, okay.
Yeah, I guess I don't know how difficult it is to, change a caste behavior. It
Yeah.
seems like that's pretty hard.
It's one protein, apparently.
Well, yeah. I
One
guess.
protein.
One peptide.
One, yeah.
Yeah, not
Is it possible to establish in the field that other things like this exist and people know this?
I mean, it might be
seems
because
like
they...
it.
Because
I
they
don't know.
say
I
that
think
this
a lot
is
of behaviors are probably somewhat conserved.
I mean, can I inject the peptide into Linea's brain and it will change her behavior?
Probably.
No, we just
She
said
might
that
forage- she might
probably
forage
the
more.
peptide injection doesn't work in big things. So you have to inject peptides into tiny
You
brains.
feed it to her
Yeah.
for 100 years.
I guess if I get a virus that expresses a peptide in my brain,
You'll go
can I
forage.
then become the
You'll
forager
forage.
then?
You'll
Yeah.
forage.
Alright,
Okay,
next figure.
now
Is
our
it the last
summary figure.
Yeah, this is the last
This is the
figure.
summary figure. Okay.
Okay, but I actually think these are...
The most important question. Did a real human being draw these?
The weird thing
I
is
think
there's
so.
no photo credit in the figure legend.
What do
Let
you
me
mean?
check the acknowledgements.
Photo credit in the figure. Alright, figure 6.
Schematic.
For leafcutter ant subcast baseline behavior and reprogramming. Is that a sentence?
No.
Not a sentence. Boo.
Baseline.
I'm okay with it.
Defense. I think that in this case,
No. I will not bend on this rule.
I do think it would be better as a sentence.
I am quite drunk and having difficulty following what's going on.
I was thinking
and
about
I
this
need
whole
a sentence
sentence,
to understand
this whole
what's going
sentence
on.
thing, and so you want it to be a statement.
I want it
Yeah.
to
The
be a
figure
statement.
legend.
So
Like,
that
what am
when
I supposed
get
to learn?
confused, I can look at what
But
the
isn't that
statement is.
inappropriate? Because this is still in the results section. So in the results of a paper, should you be making statements?
Yes.
Or should you be presenting data and then in the discussion you can make statements?
I want a statement on what I'm looking at.
Yeah.
I want some small, tiny little breadcrumb of synthesis so that I'm not completely lost. Because right now, I'm looking at this and I'm completely lost. Because there's a mole rat eating carrots or another mole rat.
Or
Well,
I think it's
I think the confusing thing here is that the schematic is for leafcutter and subcast baseline behavior and reprogramming, and somehow
There's
there's
also a naked
a
mole rat.
mole rat at the bottom of the figure.
Is a naked mole rat like is it like the traditional eusocial mammal?
I think it's
Are there
eusocial
others?
mammal.
Yeah,
I don't think
I-
there's another one.
Yeah,
that-
I don't know any others.
I guess aren't like prairie dogs sort of in. I mean, they're not. They can all reproduce, but they're very like communal living.
Okay.
So they like behave in ways that don't make sense.
Yeah, they're another mammal.
Like they'll like sacrifice themselves for the good of the colony.
Yeah. Yeah.
Chickens will do that. That make chickens eusocial.
Yeah, but they're also not mammals.
Oh, yeah, that's true.
But yeah, I mean, like even prairie dogs are like not very tractable for having a colony in your lab.
Yeah.
Okay, so this is a upper representations of each of the 4 subcasts baseline behavior are pictured.
Okay, cool.
Okay.
Middle. In
Perturbation.
majors, CCAP injection induces leaf movement, while NPA knockdown induces pupil movement.
See, he's
Pupil
holding a
movement?
leaf and a pupa.
Okay.
Pupil.
Pupil.
When I say that someone is a pupil, am I taking that word from pupa?
I don't think so.
The change in volume of us, we're really loud to quiet. This is gonna be the worst. This is just gonna be the worst.
Don't think about it, Ben.
You don't need to say that because you don't have to edit it.
Yeah, I mean, maybe I should edit them so that you don't think about
Yeah, I'm
it.
just gonna start screaming into the microphone.
Everybody editing them so that as we're recording them, you can think, oh, I leaned back for like 3 minutes and then brought my face back. That's gonna suck to fix. Lower. Transcriptomic signatures of forager and nurse subcasts in A. cephalotes are strikingly similar to those of eusocial naked mole rat H. Gilbert. This is pretty clear figure. It does bring me back to the very beginning. Why did they start with comparative transcriptomics show strikingly similarity between ant and a eusocial mammal? Like, what was the varying
I
belief?
swear it's gonna be like, I on these like highlights, you're only allowed 15 words.
Yeah.
It's just a hard pass. Like if
And
it's more than 15
eusocial
words,
mammal was shorter
then
than
naked
naked
mole
mole
rat.
rat. That's true.
3 words versus 2.
If you read this paper and we made the mistake of sending you out that we did this and you actually listened to this part, let us know if you wrote eusocial mammal because it was 2 words less than naked mole rat. I mean, 1 word less than naked mole rat.
Maybe it's like a cliffhanger, like you
Yeah.
want to know what the eusocial mammal is, so you're going to read the paper.
But we've already decided there's only one.
Well, that's
For,
true.
for those who know eusocial mammals,
11 would be a weird word.
that would be a weird cutoff. And then are any of the others longer? Some of them look
And
longer.
should it
Maybe
be
the whole
and
house?
eusocial mammal?
No, so that is it's not the it's not the letter that comes next. It's the sound that
But
comes next.
you is
U
still a
is
vowel.
still a vowel.
But you don't
Yeah,
say it.
Yeah. Maybe is.
You don't
considered
say
consonant.
so vowels are not actually
N Yuletide. A Yuletide.
I think it's
Yeah.
because of the
A.
Y.
it's about how your
A
mouth moves.
Yellow jacket. N Yellow jacket. Huh. That sucks.
It's about the
Come on, I
position
had
of your
one
tongue.
rule.
Yeah.
That sucks. I knew the like secondary rule that's not
Yeah.
what the letter actually
It's
starts
not
with.
about the letter. It's about the
but
phonetic
it's
sound.
not actually
and
about
how
the
you-
vowel. It's about the phonetic sound.
It's how
That
you produce
sucks.
the sound, and you can't say N-U. That requires you to like move your tongue
Yeah.
all the way instead of
And
N-U.
And
yellow
you.
jacket. A yellow jacket.
Yeah, it's because of the Y. Sometimes Y.
But it's not a Y.
And you
Yeah,
shall
but
show.
it's a sound of a Y.
Yeah.
N yak. A yak.
Yeah, a yak. Okay, sorry, getting us sidetracked.
Well, sidetrack to what? Like we're done, right?
Okay.
So I'll ask if you, Linnea, or anybody, no, if you could have one more experiment that they did, what would your experiment be?
Oh man.
Well, I think we discussed you wanted them to inject naked mole rats with
RNAi.
RNAi or a virus. That
I just want more
I mean,
evidence
that would be cool.
that what you've observed in ants is either conserved or convergent with what you've saying might also exist in naked mole rats. That the combo is striking and surprising and seems like it has much deeper implications. I'm not sure the best way to provide more experimental orthogonal evidence to the same claim, but that is what I would like.
Yeah, so I guess the NPA you're saying doesn't exist in naked mole rats?
Yeah, I guess it's not like a conserved peptide.
But the idea is that when you do inject it into the naked mole rats, there's some receptor.
There's
Yeah.
probably a conserved receptor.
There was like some supplemental
there
stuff
some,
that got a
like-
little fuzzy that suggested that the NPA could participate in some insulin related signaling.
Okay.
And that is probably like the orthologous mechanism.
Okay.
But again, it was very like of the same like caliber of like evidence that
It's
was shown.
presented in the main figure.
Yeah.
Yeah.
It's a great paper.
It's a great paper. Yeah, I want more naked mole rat stuff, but I think that's not the whole point of the paper, right?
That's
I
next
feel like the whole
Yeah.
point of the paper is ants and the naked mole rat.
I was wondering
and
if
that
it was one of
seems
these
like
somebody
follow-up.
told them like, okay, that's cool, it's insects. I want this to be an
Yeah.
animal.
We want charismatic
I
megafauna.
studied yeast, and we always had to be like, oh, this matters for humans too.
I guess it would be really nice to have some like you can maybe you can't do this in ants. It seems pretty hard, but like can we get some data that's not genetics? We have this behavioral data. The behavioral data kicks ass. Could we get some like neuroscience data where we have like a little probe drive where we see what neurons are firing in one case versus another? Or maybe that's just like not doable in ants. I know it's
I have an
doable.
ant farm.
It's doable in spiders, right? Which makes me think that it's not completely impossible in ants.
I do agree with your sentiment that it's like they inject the peptide or inhibit it, and then they show that the behavior changes, and that's very striking.
So this thing has died on audio
So
again.
scream.
Okay. So scream.
Scream.
No,
Just need to talk
I'm
about
just saying that thing died twice in one recording. We have to find a different solution.
Yeah, we'll
Okay,
use
that's
this
fair.
bigger thing. Why didn't we just didn't want to use it?
Well,
we
we
would
wanted
like
to,
to have a solution where we can go to an actual pub and
I just
do
think
this
we
the
can bring
way
that thing.
you think we're gonna bring the whole. It needs to be plugged into an outlet.
Well then we can't.
But also we could try two mics, and maybe it would last long.
Yeah, we could try 2 mics. I think the next time we do this, I'm gonna try clip-on mics. Is what I'm gonna do and see how bad the audio is.
Mm-hmm.
Does that mean every person has a...
Yeah,
Your own
that's
battery?
fine.
Every clip-on mic should have a certain amount of time it lasts. We'll find out. And then after we do that, we'll go to a bar and we'll see just how bad the bleed through on clip-on mics is. Anyways, I think that's it for us tonight. Unless anyone has any last words they want to say.
Great paper.
Great paper.
Love the graphics.
Okay. And I guess maybe Megan and Zach are reading these papers from now on to tell us what's up.
I'm not committing
No
to that.
pressure.
High bar. Alright, thanks. Bye.